and thank you for standing by. At this time, all participants are in a listen-only mode. This call is being recorded. If you have any objections, you may disconnect at this time. I will now turn the meeting over to your host, Laura Bleacher. Laura, you may begin when ready.
Good morning. I'm Laura Bleacher from NASA's Office of Communications. Thank you for joining us today to talk with the experts about science experiments launching on Northrop Grumman's 18th commercial resupply mission for the agency to the International Space Station. Northrop Grumman is targeting Sunday, November 6, 2022, for the launch of its Cygnus spacecraft on an Antares rocket from Wallops Flight Facility in Virginia. Here to talk with us about some of the research launching aboard the Northrop Grumman Cygnus are Heidi Paris, Associate Program Scientist for the International Space Station Program, who will discuss the breadth of research being done aboard the orbiting laboratory, Dr. Annalisa Paul, Principal Investigator for Plant Habitat 03 and Research Professor at the University of Florida, Gainesville, Dr. William Ulbricht, Acting Deputy Division Director for the National Science Foundation's Division of Chemical, Bioengineering, Environmental, and Transport Systems, who will be giving an overview of the many NSF payloads on the flight, Dr. Ingrid Tomatsa, Principal Investigator for the Catastrophic Post-Wildfire Mudflows Experiment and Professor at the University of California, San Diego, Dr. Andrea Fuso, Co-Principal Investigator of the OvoSpace Project and Associate Professor at the Sapienza University of Rome, Rich Bowling, Vice President of Corporate Advancement for In-Space Manufacturing and Operations for Redwire Space, who will be discussing the reflight of the biofabrication facility, and Timothy Kudzanaye Kuhamba, Senior Systems Engineer at the Zimbabwe National Geospatial and Space Agency, who will discuss the BIRDS 5 project. Our speakers will provide a brief overview of their research, and we will open the lines for a few questions after each speaker. If we have time at the end, we can take additional questions. Your phones are on mute now. To get into the question queue, you can press *1 on your phone at any time. We will call on you and the operator will open your mic so you can ask your question. We ask that you please stick to one question. If we have time, we'll allow reporters to ask a second question. We'll also be taking questions using the hashtag AskNASA on social media. About 1 hour after we conclude, you can listen to a replay of this teleconference by dialing toll-free 866-361-4941. That's 866-361-4941, or by visiting the NASA Videos YouTube channel. To start, we will hear from Heidi Paris, Associate Program Scientist for the International Space Station Program and Research Integration Office Deputy Manager. Over to you, Ms. Paris.
All right, great. Thank you, Laura. Good morning, good afternoon, everybody. Thank you very much for joining in today to discuss the research that's going to be launching on NG-18 next weekend. We are extremely fortunate today to have with us several of the science teams behind these experiments. So we're very much looking forward to hearing them discuss their science later in the telecon. First, I wanted to start out with just an update on the state of ISS research and also a very quick overview about some of the science launching on this flight. This is a really exciting time for research on board the International Space Station. After over 2 decades on orbit, International Space Station is really hitting its stride with dozens of state-of-the-art laboratory facilities that are on board and being used to perform hundreds of different and new experiments every year. And that research spans every major scientific discipline. We also see indications that results from ISS research are increasingly starting to make their way into our everyday lives in the form of new and better products and technologies, breakthroughs in the development of new medications, new medical technologies, better data to study how Earth's ecosystem is changing, and also helping to solve the challenges that are going to enable long-duration space exploration outside of the low Earth orbit. Now to talk a bit about the research launching on NG-18. So this vehicle is going to bring up approximately 800 kilograms of hardware and samples to support right around 40 different scientific investigations, 20 of which are brand new studies in areas like physical sciences, technology development and demonstration, and biology and biotechnology. In addition to the science, NG-18 is also going to be carrying crew provisions and hardware for space station upgrades, including some hardware that will be used to support installation of new solar arrays, which will literally give us the power to do more science. We have several investigations on this flight, including OvoSpace and Plant Habitat 03, that are seeking to improve our understanding of biological processes in the spaceflight environment in order to help us thrive as we continue to explore further into space. We also have several investigations, including the BIRDS 5 project and catastrophic post-wildfire mudflows investigations. These are going to be using ISS in different ways to enhance our understanding of either environmental processes or to provide observations of regions of interest on Earth that don't necessarily have a lot of existing data available. And then we have the biofabrication facility, and it's seeking to take advantage of the sustained microgravity environment in order to develop products that are superior to what we're able to produce on Earth. So we'll hear more about each of these during the telecon today, and I don't want to take up any more time, but I do want to leave you with a couple of references for more information. If you'd like to hear more about how ISS research is benefiting humanity, we have recently published an update to the ISS Benefits for Humanity book, and that can be found at nasa.gov/station benefits, all one word. And also, you can follow along with up-to-date on-orbit research activities by following us on Twitter at ISS_research. All right, Laura, back to you.
Great, thank you, Ms. Paris. Now we will hear from Dr. Annalisa Paul, Research Professor at the University of Florida, Gainesville, and Principal Investigator for Plant Habitat 03. Dr. Paul is the Director of the Interdisciplinary Center for Biotechnology Research at the University of Florida and a professor in the Department of Horticultural Sciences. In the past 20 years, Paul and her colleague Rob Furl have launched 11 orbital experiments to study the effects of the spaceflight environment on the molecular genetics of the model plant Arabidopsis to help understand how plants physiologically adjust to this novel environment. Dr. Paul?
All right. Thank you very much, Laura. So first of all, the name of our experiment, we call it Plant Habitat 03, doesn't tell you much. It just means that that's the third experiment to launch to this particular type of habitat that is resident on the space station. And why this habitat is unique is that it allows plants to grow large. Previously, in all our other 11 experiments, they've just been growing on Petri plates to look at their molecular genetics and their adaptation. But now what we're going to do is in this experiment, and the name of the experiment is Epigenetic Adaptation to the Spaceflight Environment. And we're going to be looking at genomic change that's induced over generations in space. And so we needed a habitat that would allow plants to grow to maturity. Now, Arabidopsis is small and grows very fast, and so we can actually get a full generation of plants in this reasonably small habitat on the space station. And what we're going to ask is, how do the epigenetic effects that happen in the adaptation to spaceflight, how are those changing the way plants respond in their patterns of gene expression, and how they might be transferred to the next generation. And this is the exciting part, is because we know that on Earth that plants will respond to environmental stressors like pathogen attack or heavy metals in soils and such, so that their progeny are actually better adapted to those stresses than their parents were, not because they're transferring magic genetic code in a Lamarckian way to the next generation, but because their DNA is modified by the stressors to allow different portions of the genome, different portions of the genes, to be more or less accessible to the types of stress responses that those challenges elicit. And those modifications, it's sort of like the decorations to the genome, which are called, in our case, methylation, then are transferred to the next generation, the next progeny of plants, and are sort of primed to be able to respond to those kinds of stressors more quickly, more effectively than their parents were. And this is what is exciting for us, to think that maybe one generation of plants on the space station would then sort of predispose their next generation to be better adapted to spaceflight.
Thank you. We will now go to reporters for questions for Dr. Paul. Please press star 1 on your phone to be entered into the queue. And while we are waiting for reporters to put in their questions, I'm going to use moderator privilege to ask one. So can you tell us a little bit more about why it is important to look at the molecular biology of these plants? What can the gene expression patterns tell you that you could not discern from just studying how they were growing in space?
Oh, absolutely. And so when you look at the molecular genetics of a plant and the patterns of genes, what turns on, what turns off in response to an environmental stress, it gives you a look into the toolbox, the metabolic toolbox that plants use to adjust their metabolism to adapt. And so I liken it to you go into somebody's garage and you see what tools they left out on the floor, and you can tell by that set of tools what was important to that project. If it's saws and hammers, it was carpentry. And so same thing is true about the patterns of gene expression. If they're turning on genes that are associated with, say, cell wall remodeling or reactive oxygen species, you can tell that those aspects of their metabolism are important to adapting to that environment and what's important to those plants. And once we learn what those tools are being used, it helps us then either engineer plants themselves or engineer their habitats to make some of those stressors less impactful, or to choose in the future different types of crops that are better suited for dealing with those types of stress responses.
Great, thank you. If there are no other questions for Dr. Paul, then we will move on to our next speaker. Next, we'll hear from Dr. William Ulbricht, Acting Deputy Division Director for the National Science Foundation, Division of Chemicals, Bioengineering, Environmental, and Transport Systems, who will be giving an overview of the many NSF payloads on the flight. Dr. Ulbricht has been a leader of NSF's partnership for engineering research aboard the International Space Station National Lab. Dr. Ulbricht?
Thank you very much, Laura. As you probably know, NSF is one of the major funders of basic research in the U.S., and NSF Engineering has supported research projects on the ISS since 2015. And we have 7 projects on board this mission, and we're very excited about it. The program that we run emphasizes projects that benefit life on Earth, and people who propose experiments for our support must present a really compelling argument about how the experiments in microgravity can produce results that benefit engineering on Earth. And the ISS provides a unique facility for our research community with sustained microgravity, and this is not available to our researchers in any other way. As Heidi mentioned, we have projects in both the physical sciences and biological sciences on board this mission. Many of our researchers study the behavior of complicated fluids, for example, liquids that contain particles or gaseous bubbles. These kinds of systems are relevant to a wide variety of processes on Earth, and they're involved in the production of products ranging from new building material to cosmetics, foodstuffs, drug delivery vehicles, and others. And they're also relevant to naturally occurring processes, which we'll hear in a minute from Dr. Tomac. On this mission, we have experiments studying a variety of processes, including bubble coalescence in foams, the bubble formation on boiling surfaces that affect heat transfer. And in microgravity, we're able to remove the effect of gravity and see the effects of other forces that are sometimes hard to reveal on Earth. So the results of these experiments can help us deduce the roles of these forces on various systems of interest. They provide new information about the physics, the chemistry, and the biology of these systems that we can use to optimize processes that take place here on Earth. Two of our experiments on this mission focus on cellular systems, one involving osteoblasts, these are bone-forming cells, and how their mechanical properties influence signaling pathways that influence their growth. And the other project that we have is focusing on the growth and maturation of cardiac muscle cells to enhance their large-scale production for use in regenerative medicine. So it's a variety of projects, and we're very happy to be on board with this mission. Back to you, Laura.
Great. Thank you, Dr. Ulbricht. We will now take questions for Dr. Ulbricht. Again, please press *1 to be entered into the queue. Dr. Ulbricht, can you talk a little bit more about the application of knowledge from experiments that are done in microgravity and then applying them on Earth where there is gravity?
Yeah, that's a good question, and it's a challenge for our investigators. But in many cases, as I mentioned, there are forces on Earth that are obscured by the effect of gravity. These forces are important, but they're difficult to study in a small-scale experiment because gravity is such a large influence when you do a laboratory experiment on a small scale. For example, I mentioned foams. You can imagine that bubbles in a liquid would rise quickly, and they do in a small-scale experiment on Earth, and that pretty much ends the experiment. But in microgravity, the bubbles will remain in suspension, and we can study the detailed dynamics that expose the role of other forces. And then these results can be placed into models that become predictive on Earth about the behavior of these materials. So we're really discovering the underlying physics that are tough to see on Earth and incorporating those results into models that can help us in engineering design.
Thank you. As a reminder, we are also taking questions on social media using the hashtag AskNASA. And if you are a reporter on the line, please press *1 to enter the question queue. Joining us next is Dr. Ingrid Tomatsa, Principal Investigator for one of the NSF-sponsored payloads aboard NG-18, catastrophic post-wildfire mudflows. Dr. Tomatsa is a professor at the University of California, San Diego. Over to you.
Thank you so much. So I will talk about my project, which is composition and flow behavior of post-wildfire mudflows. First, I would like to tell you that we must understand the effect of gravity on post-wildfire mudflows for three main reasons. First, wildfire frequency has been exponentially increasing in past years, and after wildfires, burned scars are vulnerable to rain events, where catastrophic mudflows blanket down bare slopes and destroy nature, infrastructure, and communities under hills. Second, post-wildfire mudflows differ from regular rain-induced mudslides and debris flows because they can develop and flow very fast downhill under gravity, provoked by relatively low raining intensities of about 15 millimeters per hour and very short rain durations like 10 to 15 minutes. Third, wildfires exposed burned hills to rain because the vegetation cover is gone. Also, the topsoil surface turns water repellent due to the deposition of organic matter combustion chemicals on soil's particle surfaces. So, water repellent particles interact with air bubbles in an exciting way that cannot be fully understood only in gravity conditions like our laboratories on Earth. Water repellency that impacts granular soils like sand changes the water particles' behavior. As a result of water repellency, soil particles react with air bubbles. Consequently, post-wildfire mudflows comprise of rainwater, soil, and some trapped air in form of those bubbles inside the mixture. The history of the issue is that general slurries of water and soil particles would not be able to transport substantial rock boulders as was observed in Montecito in 2018, and would accordingly not be able to flow so fast and produce such large impact forces, destroy houses, and even take lives. But if we consider that mudflows contain some amount of trapped air, a mixture of bubbles that are trapped inside and particles in water could be very different, and it could carry heavy debris, right? So if you look a little bit closer what is going on and what we are trying to research on ISS, in fact, that air gets trapped in the mixture by water repellent particles that cover different air bubbles. So in gravity, air tends to rise and would exit the mud flow mixture unless trapped. On the contrary, soil particles, such as sand, are solids, and they tend to sink to the bottom in water, for example. So as you can imagine, gravity plays a significant role in the internal structure and behavior of a post-wildfire mud flow because of those hydrophobic or water repellent particles that would trap the air bubbles. Those agglomerates may sink or they may float in case of tiny air bubbles trapped inside larger particles. And we want to understand better what forms air trapped in water and how the water repellent soil particle and air form different shapes like pipes or cylinders and spheres. In order to do this, we will compare results for laboratory experiments in Earth gravity with microgravity conditions, predominantly to quantify the baseline for particle bubble attachment. But in addition, we want to understand the role of mixing energy, size of particles, relative volumetric ratios of three phases that we are observing, solid, water, and air, on the mixture internal structure relative to gravity. The final goal is, as Dr. Olbricht highlighted, is to understand the forces better in these mixtures that exist when gravity is removed. So when this very strong effect of sinking of solids or lifting and rising of the air is removed. And we hope that we will be able to develop some better formulas for fluid and mixture flow and transport that will enable us better modeling of mud flows, post-wild mud flows, and also predicting of the spread and impact that post-wildfire mud flows may deliver after the burn and before the rain.
Thank you. We will now take questions about the catastrophic post-wildfire mudflows experiment. Again, please press *1 to be entered into the queue each time. One question we have is, what areas of the world could these results most benefit?
Generally, wildfires are increasing all over the world. So we have a lot of new wildfires and mudflows in California, but in Mediterranean regions they're reported as well. And any other climate that is impacted by climate change and also by heat that causes more wildfires, and then there will be rain all over the world, I would say.
Great, thank you. We also have a question from #AskNASA on social media. Rob is Hungry on Twitter asks, why study mudflows in space?
We would like to better understand how the trapped air in the post-wildfire mud flow connects to sand particles. There are mudslides and mud flows that occur in nature under very heavy rain. These normally don't contain those water-repellent hydrophobic particles. And we know a lot about mud flows. I'm geotechnical engineer, and this is a branch of civil engineering. We have been studied mud flows for many, many years on Earth. What is different with post-wildfire mud flows is the water repellency of the surface of particles, which occurs due to the burn of organic matter, which has been known for a decade now. And these water-repellent particles are trapping the air inside the mud flow. So this new component of air has not been well understood for the internal composition of the mud flows.
Thank you. We'll now hear from Dr. Andrea Fuso, co-principal investigator of the OvoSpace experiment, for which will take care of the epigenetic analysis as well as the coordination of the team preparing the experiment to be launched. Dr. Fuso is also an associate professor of clinical biochemistry and clinical molecular biology at the Department of Experimental Medicine, Sapienza University of Rome. Dr. Fuso?
Yes, thank you. So OvoSpace is funded and coordinated by the Italian Space Agency, ASI, and it is a collaborative project between our department at Sapienza University of Rome and AELI, an aerospace company based in Naples, Italy, and Nanorex as implementation partner. AELI realized the Minilab, which is the hardware that will host our cells during the flight and will guarantee the correct cultural conditions. So the project is aimed at studying how ovary cells, specifically granule cells and theca cells, respond to microgravity. The cells surround the oocyte and are primarily responsible for sustaining oocyte maturation, mainly through the secretion of steroid hormones, and are therefore directly involved in fertility. The cells will be seeded in the chambers of the Minilab that will provide injection of fresh culture medium after the docking to the ESS. Then, after 72 hours, the Minilab will inject a fixative that will preserve the cells until the return on ground. We will use the cells to profile the steroid hormone secretion and the molecular processes that regulate steroidogenesis, respect, of course, to the cells cultured on ground. And, of course, we can send only a limited number of cells on the ESS due to the limited space in the Minilab. Therefore, we will take advantage of a random positioning machine, RPM, in our lab, which mimics the microgravity to work on a larger amount of cells. These will allow us to perform a larger screening and to refine the analysis on the cells sent to the ESS. We will study hormones production and secretion, as well as the molecular mechanisms regulating the expression of the genes related to steroidogenesis. This will include epigenetic analysis as well. Epigenetics consists in different molecular mechanisms that drive the level of expression of our genes in our body. So we think that this study will clarify the fertility potential in microgravity, highlighting possible alteration in steroidogenesis, and suggesting possible treatments that can improve fertility if necessary, preserving ovary cell function and therefore preserving fertility. Of course, this result will likely give us also useful basic and translational data to support fertility on ground. And as far as we know, this is also one of the few experiments analyzing epigenetic alteration in mammal cells on the ESS. Thank you.
Thank you, Dr. Fuso. We will now take questions about the OvoSpace investigation. Again, please press *1 to be entered into the queue, or use the hashtag AskNASA on social media. I see we have a question in the queue.
Our first question comes from Elizabeth Howe, space.com. Your line is open.
Hello, thanks for taking my question. I wanted to get some more details about how you would be learning more about the fertility of these cells and then possible next steps after this experiment, assuming that everything goes well.
Yes, this is probably the first time we work on cells related to fertility and to reproduction on the ISS. So this is really a first approach, and it is for the moment an observational study. We will study how the steroid hormones that are necessary to support the development and the maturation of the oocyte will be affected by the microgravity. So we will study cell morphology and hormones production at protein levels and hormone secretion outside the cells, and also the molecular mechanisms responsible for the regulation of gene expression, particularly of that genes involved in the hormone production and maturation and secretion. So at this point, we are aimed at assigning the base level of the study and see whether the steroidogenesis is affected by microgravity. Then we will start to reason whether we can act by supplementation of drugs or nutraceutical compounds to improve possible alteration of steroidogenesis.
Thank you. Next up, Rich Bowling will be discussing the reflight of the biofabrication facility. Mr. Bowling is the Vice President of Corporate Advancement for In-Space Manufacturing and Operations at Redwire Space. Mr. Bowling?
Thank you, Laura. On behalf of everyone at Redwire, I'm eager to share some details with you about the return of our 3D bioprinter to the International Space Station and what we're preparing to do with it over the next few months. Redwire is a global leader in space infrastructure. We provide the foundational building blocks that are enabling the most complex space missions. From power to payloads and structures to software, our innovative solutions, subsystems, and components are the critical mission-enabling technologies that make possible some of the most impactful space missions and operations of today and the future. Now, about the size of 3 microwave ovens, small ones at that, the Redwire Biofabrication Facility, which we just like to call our BFF, was the first American 3D bioprinter flown in space when it arrived at the ISS in July 2019. We brought it back to our lab in Indiana the next spring to add a few new capabilities, such as the ability to finally control the temperature of each print head. And now we're excited to see it launch aboard NG-18 for what is expected to be a long-term stay aboard the station. Besides BFF, we're also launching a veteran Redwire payload called the Advanced Space Experiment Processor and some supplies, all in preparation for bioprinting a human knee meniscus for our customer, the Uniformed Services University 4D BioCube Center for Biotechnology. The meniscal injury is one of the most common orthopedic injuries affecting military service members, and this Defense Health Organization is exploring how printing in microgravity may provide unique advantages for tissue engineering. Now, this space-printed meniscus won't be transplanted into a patient, rather it'll be studied in a lab following its return to Earth. While that is a goal for which we and our customers are striving, and while we will be making blood vessels and cardiac tissue on other missions later next year, an important immediate use of BFF is for drug efficacy testing. In space, we can print and culture organoids, which are miniaturized and simplified versions of an organ. Researchers can then test new drug compounds on these organoids. This can help reduce the need to use laboratory animals, and because the organoids can be printed with human cells, the results will be more applicable to human health outcomes. I believe the versatility of BFF will keep it in high demand among institutional and industrial researchers in myriad fields for years to come. I'm approached nearly every month by someone proposing a new use for it, with one of the most interesting lately being a way to manufacture cultured meat as an in-space food source. Following the launch of NG-18, Redwire will have 9 of its own life and physical sciences devices aboard the ISS, more than any other company, with all of them available for commercial, academic, and government partnerships in in-space research and manufacturing. Through our steady cadence of technology development and research aboard the ISS, Redwire has developed a catalog of equipment and services that make it the preferred outfitter for new Earth-orbiting space stations such as Orbital Reef and for destinations in deep space. And so on behalf of everyone at Redwire, we first want to thank the ISS National Lab and NASA for their support. We're excited about the launch of NG-18, and we say go Cygnus and go Redwire.
Thank you, Mr. Bowling. We will now take questions about the biofabrication facility. Again, please press *1 to be entered into the queue each time, or use the hashtag AskNASA. And in fact, we have a question from Twitter from Michael Petch. Can you say more about the roadmap or time frame for scale-up of BFF operations? When might transplantable organs made using the biofabrication facility be available, and what challenges are there?
So I think to cut to the chase, I think perhaps by the end of the decade we'll see transplantable tissues for in lab animals. It's going to be a lot further into the future for human beings. The regulatory process alone could easily take a decade or more. But the roadblocks really are those that we're knocking down with every mission that we see. We're making a lot of great new progress with every mission. I think it's a matter of time. We are striving to achieve that goal, and we're hoping to do a lot of good science along the way as well.
Thank you. Finally, we'll hear from Timothy Kudzanaye Kuhamba, who will discuss the BIRDS 5 project. He is a senior systems engineer at the Zimbabwe National Geospatial and Space Agency and holds bachelor's and master's degrees from the National University of Science and Technology and the Kyushu Institute of Technology. Mr. Kuhamba?
Thank you very much. The BIRDS 5 satellite project has got 9 missions in total, of which 8 are the same for all the nanosatellites, and only TACA has the particle instrument for nanosatellites, which is the PINO. So we've got the missions of the land use land cover, water quality assessment, soil fertility, store and forward, birds nests, altitude visualization, onboard image classification, altitude determination and control. So for the land use and land cover, the satellite images that the multispectral takes the pictures of the participating countries, Zimbabwe and Uganda. And these are used to investigate land use and land cover, such as crop yield, soil adjusted vegetation index, cultivated area, as well as the leaf chlorophyll index. The cameras are also used to take images for the assessment, for the water quality assessment, which will take images of the dams and the lakes, and they'll be used for the demonstration for the water quality assessment. So this is very much important in helping Zimbabwe and Uganda in attaining the SDG 6 goal. We also have got the mission of soil fertility, in which the cameras are used to capture the images of the member countries, and these images, they facilitate the analysis for the nitrogen, which is the nitrogen in the soil, which will be used to analyze whether the soil is good for agriculture. Then we also have got the onboard image classification, which the satellite classifies the images based on the machine learning algorithm, and it sends the results to the ground. It's to improve the efficiency of the data of the downlink process, and a larger CubeSat data set can be captured for future machine learning algorithms. We also have got the store and forward mission, which demonstrates the data and message in line with the universal amateur radio text. So the store and forward mission has got 2 primary activities: to collect the solar illumination measurements for remote sensors and forward the data to the ground, and also to collect the data related to possible disaster predictions such as landslides. I can say that in March 2019, the Southern Africa was hit by a tropical cyclone, which was rated as the worst ever experience in the continent. So this really affected people in Zimbabwe, of which 270 people were left homeless and 341 were dead. So for a country like Zimbabwe, this entails that the mission will improve the level of preparedness as this will assist the alert of the epidemic committees in making some timely decisions. Then we also have got the Bird's Nest mission, which is a phone application for displaying the data of the bird satellite. The Bird's Nest is free, providing knowledge and raising awareness about space around the world. It can be found on the Google platform and also the iOS. We also have got the satellite outreach visualization by getting the magnetometer data, the gyroscope, and the solar currents. This can be used to visualize how the satellite is moving in orbit, as well as to assist the capturing of the images of the targeted areas in order for the land use and land cover and the water quality to be executed well. We also have got the particle instrument for nanosatellites. This is a scientific mission. So we've got the PINO, which will measure high energy electrons precipitating in the upper atmosphere and will contribute to the monitoring of the space weather at low Earth orbit and to understand the loss of the radiation belt electrons and the influence on the upper environmental. So these are the missions for the BEDS 5 project. And I would like to thank the Zimbabwe National Geo and Spatial Agency, the Kyushu Institute of Technology, as well as the Ministry of Higher and Tertiary Education, Innovation, Science and Technology from Uganda. So thank you very much for listening.
Thank you. We'll now take questions about the BIRDS 5 project. Again, please press *1 to be entered into the queue or use the hashtag AskNASA. I'll use moderator privilege to ask a question about what was the greatest challenge in developing the BIRDS 5 project?
Okay. The challenge was that more than ever, because the satellite development was in the pandemic. So the satellite development process started in April 2020 and ended in March 2022. So there was a restriction of many activities, especially the things that needs to be done in person. So the problems that can be solved simply by talking to each other were now very much difficult through texting and online meetings. So the problem, for example, if one has got a fever, he goes into self-quarantine, and that work needs now to be done virtually. So this will also make the project be lagging in terms of the project timelines. So this really affected the project, but however, working as a team, we managed to overcome this problem, and we managed to finish the satellite development in less than 2 years.
Thank you. We will now take additional questions for any of our speakers. Again, if you would like to ask a question, please press *1 on your phone, and please identify to whom you are addressing your question. While we're waiting for questions to come in the queue, I will ask another question from hashtag AskNASA. I believe this is for Mr. Bowling. This question is from Keisha Rogers TX on Twitter. How will printing organoids with human cells help with transplants of organs? Would you use the same cells of an individual to create an organ regenerated from their cells to help alleviate rejection? Mr. Bowling, you may be on mute. All right, we will take another question on Twitter, or from Twitter. This question is from Ashley B470455128, who asks, how long does it take to get to the ISS? Heidi, are you able to answer that question for us?
Yeah, I can take that one. So the time that it takes to get to ISS really depends on a number of different factors, and it's different for every flight. So we have to look at things like the phasing window between the launch site and the International Space Station, and the different energies that are responsible to get to that orbit. For NG-18 specifically, let me see if I can find that information. You know, we don't have that information available right at this moment, but I can get back to you later in the telecon with that information.
Great, thank you. We also have another question from social media. Sakria3226631 on Twitter asks, whenever I hear about science in space, I always wonder what the differences are between plants grown on the ISS and plants grown on Earth. We'll direct that to Dr. Paul, please.
Hi. So the difference between plants growing on the ground versus plants growing on the ISS. We've done a lot of different experiments that look at the molecular biology of things. And so there are two sort of aspects you can look at. First of all, the way plants grow is that normally plants use gravity, of course, as a cue as to what grows up and what grows down. And we found that on the ISS, plants tend to use light as a cue as to which way to grow their roots. Because if you think about it, if they had no sense of which way to grow, they would just germinate and grow in these little tight balls where they would use up all their resources all at once. But they don't do that. And the first thing we noticed is that plants use light to show them which way to grow away from. And so then plant roots grow away from a light source, and that allows them to sort of explore the medium in which they're growing. But in addition, when we did some follow-on experiments of growing them in the dark or growing them without a directional light source that would give them that cue, we also found that there are inherent mechanisms in plants that allow them to grow away from where they're planted by doing a sort of a spiral random walk away from the seed germination point. And so plants on the ground also have this mechanism, but until we took gravity out of the equation, until we took gravity away so it was no longer influencing how plants grew, we never knew that plants had this inherent mechanism of how to navigate away from their planted. So the bottom line is that plants grow really just fine without gravity. It's just that you have to sort of help them with directional cues to maximize how they get nutrients from their surroundings, whether that be from porous materials that are watered by capillary action or on a gel matrix like we often grow them. But the bottom line is they do pretty fine, but they turn on a lot of different genes that help them adapt to that unique environment.
Great, thank you. We have another question on social media. I'll direct this one to Ms. Paris. How will future Artemis missions help us further educate ourselves with how extended studies in space will affect the human biology and psychology? And I believe you also may have an answer to us about the transit time to the ISS.
Sure, I can start with the transit time. It's going to take roughly 48 hours for this mission to go from launch to berthing with the International Space Station. And now to talk about the Artemis question. So, of course, we're very excited for the upcoming launch of Artemis 1, which is targeting mid-November. The International Space Station really has been used as a test bed along the way for a number of the technologies that will be incorporated into future Artemis missions, as well as the science that's going to be flying actually on this Artemis mission as well as in the future. Specifically, we have a science investigation flying on Artemis 1 that is aimed at providing a better understanding of how the radiation environment will affect yeast cells outside of the Earth's direct orbit. And yeast cells are very similar to some of the cells in our bodies. So that mission specifically, that science investigation was based on some precursor studies that occurred on the International Space Station back in the 2017 timeframe. So a lot of uses there of cases where we're using ISF as a test bed to inform future science on Artemis missions. And also there are a number of technologies that we're testing out now on the International Space Station that will be used to inform the design of future Artemis systems, including things like SURFI, which is a new spacesuit technology, and also especially our ECLIS systems, environmental control and life support systems, where we're looking to really be able to close the loop and recycle almost all of our air and water, which is extremely important for future long-duration space exploration.
Thank you. If there are no more questions, we will wrap up today's call. Thank you for your interest in this important science, research, and technology launching aboard Northrop Grumman's 18th commercial resupply mission for NASA. For imagery of these investigations, please refer to our NG-18 Science Highlights feature on nasa.gov/ISS-science. You can follow all the research being conducted during the Expedition 68 mission by visiting nasa.gov/station-research-news or following @ISS_research on Twitter. Stay tuned to nasa.gov/Northrop Grumman for regular updates in advance of the upcoming launch. We are targeting Saturday, November 5th at 1 p.m. Eastern Time for our pre-launch news conference. Launch broadcast coverage will begin at 5:30 a.m. Eastern Time on Sunday, November 6th. As a reminder, you can listen to a replay of this teleconference by dialing 866-361-4941. Again, that's 866-361-4941, or by visiting the NASA video channel on YouTube. Thank you all for joining us. This concludes today's telecon.